sliding over one another. So that's the thick one going across there, that's the thin one, and then forming these cross bridges where the thick touches the thin. We'll do that in a bit. So that's again a very nice electron microscope, a very nice neat one of a muscle fibre. Some key terms that you don't need to memorise but just to be aware of. Sarcoplasm is the cytoplasm of a muscle cell. Sarcolemma is the membrane of a muscle cell. Sarcoplasmic reticulum is the endoplasmic reticulum of a muscle cell. So you might have met those cytoplasm, endoplasmic reticulum in your cells. So muscles just call it sarco. So sarcolemma, sarcoplasm, sarcoplasmic reticulum. Also, we've said that muscles, to be able to contract, need lots of ATP, which is made by aerobic respiration in the mitochondria. So there's loads and loads and loads and loads of mitochondria in a muscle cell. So that's kind of what the edge of that diagram is showing. So loads of mitochondria, and then we're getting onto the proteins. So you can see these thick bands, and these black bands running down it. They are called the Z lines. Oh, don't worry, I'm going to show another image with it all on. And Z line to Z line is one sarcomere. I'm warning you, there's so much terminology, you're going to hate me. There's going to be like 20, 30 new words in the next 10 minutes. So please try to bear with me. So that's a muscle, one of those muscle fibres down the electron microscope. So do you know the stripe pattern you saw?
Yeah.
Yeah, that's that.
That's one of them little stripes.
Yes. Well, not one of those stripes, well, that would be one stripe.
Right.
Yeah, and that would be another stripe. Yeah? So what you've just seen down the microscope, you saw those stripes. That's one stripe, that's another stripe, that's another stripe. Everybody okay now? Yeah? So, stripe to stripe, the proper name for the stripe is called the Z line. So kind of A to Z, it's at the end. And Z line to Z line is how the muscle contracts. So when the muscle contracts, these Z lines are going to get close together. And then these two will get close together, and these two will get close together. And the entire muscle is going to kind of move in. So if you saw the muscle contract, those lines will get closer. Actually, I'm going to just take these off and find another image. Sorry, I just want to strengthen muscle. Right, let's finish. I'm just going to show this video because it might help you see what these Z lines are.
Muscle contraction. Mecanonated rabbit muscle. In this preparation, which is stored in glycerin, most of the cellular proteins are removed, leaving the contractile proteins actin and myosin.
So they've got some rabbit muscle, taken all the other bits away, and left the bits that contract. So the actin and myosin, those thick and thin filaments. It'll make sense in a second at the end.
The delicate muscle is supported by a wooden stick. The muscle fibers can be teased apart from each other using fine forceps. In this video sequence, the investigator is separating several muscle fibers and will then isolate a single muscle fiber using the stereo microscope.
Stereo microscopes is what you can see what you're doing under. So it's like what you've used sperm under as well, you can kind of see the move.
After isolation, individual muscle fibers can then be viewed at higher magnification using a compound microscope.
So that's just to zoom in so he can see what he's doing before it goes on to a high magnification microscope.
The regular organization of actin and myosin causes the striated...
So that is Z line, that's a Z line, that's a Z line, that's a Z line. So that's what you've seen in your image. That's what I was trying to find to get for you. So that Z line to Z line, the name for it is a sarcomere.
So on that other one you showed us, those ones that look like that are those
Yes.
in the middle there?
Yeah.
Right.
Yeah. So that's why I wanted to get this up, just so it made sense for what you've just seen.
Yeah.
So when a sarcomere is the smallest contractile unit. So when the muscle contracts, and this guy in a second is going to give the muscle something called ATP, so it's going to give it the energy it needs to contract. It's going to basically make the muscle contract outside of the rabbit's body, is what he's going to hopefully do. And you'll be able to see, hopefully these lines will get closer together, so the sarcomeres will get closer together. Fingers crossed.
Appearance of the muscle when viewed at higher magnification. The contractile proteins remain sensitive to the energy-carrying molecule ATP. When a solution of ATP is added to the muscle fiber, it contracts. This demonstrates that ATP alone can cause
So that's showing you that muscle contracting.
contraction of actin and myosin, leading to muscle
So that's
contraction.
just one fibre, and there's
The muscle
thousands of these that
fiber
make the muscle.
contracts. This demonstrates that ATP alone can cause interaction of actin and myosin, leading to muscle contraction.
So, and then think of that on a bigger scale, so we're on a microscopic scale, thousands and thousands of those inside the muscle. So if we go back to this now, so we've got Z line and Z line, which is making up something called a sarcomere. So when a muscle contracts, these sarcomeres all get closer together, the whole thing kind of moves in. If you think of it with a concertina and just pushing that in. So when you get an electron micrograph, you need to be able to say about the different bands. So Z lines are two that you'll need to identify and say that's a sarcomere. The one in the centre, the middle, is called the M line. So it's another line that runs down the middle, so that is the M line. And this is what's on that sheet. I've given a handwritten A3 sheet at your table. Excuse my handwriting, I did my best to try to make it neat. So this is kind of a zoomed in version now, so we're zooming in even more. So that was lots of sarcomeres. So we've got sarcomere, 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 sarcomere. And that is zooming in to one sarcomere. That one there. Yeah, no? Mm-hmm. Yeah? Okay. So we can see the Z lines. And when the muscle contracts, the Z lines move in, the muscle gets smaller. And then you can see the M line in the centre. We've got other parts we need to label on this muscle now. I'm just going to rub things off so I can draw. So that's an M line, so Z line, and that's your M line in the centre. Then you've got this band called the I band, which stays a lot lighter. And the I band is where the thin filament is. And the thin protein is called actin. So the actin is attached to those Z lines, and you find that within the I band. And that is the thin one, so it appears lighter. Okay, is that okay still? Mm-hmm. In the centre bit here, that is called the H zone, and you only see the H zone in a non-contracted muscle, so when it's relaxed. Sorry, so you only see the H zone in a relaxed muscle, and the H zone is this thicker protein called myosin, which is anchored to the M line. So that would be the H zone, which is kind of a pale grey colour. The final label is the A band. And the A band is this darker section. So the H zone lies in the middle of the A band. So the H zone is part of the A band. And that is where the two overlap. So this thick filament, this myosin, extends, and where the actin and myosin, where the thick and the thin overlap, sorry, I'm just kind of finishing the diagram. It's not the neatest ever, but you get the idea. So where they overlap, that is the H zone. So if we're just labelling the one I've just drawn on the board, you've got the I band, which is only the thin. You've got the H zone in the centre, which is only the thick. And then you've got the A band, which is the thick to the thin. So it's where they overlap. It is written on the sheet here. It is written down, so I want you to listen rather than have to worry about writing loads. So, kind of looking at this one here, you can see the thick, see the thin, and you can see where they touch each other in a second. I've put that kind of diagram on your sheet as well. So I've tried to, I do this by cut and stick, so I've tried to kind of print it to the same size and stick it so it matches up. So you can see, you've got the thin, you've got the thick, and then you've got the A band where the thick and the thin overlap. So I'll come on to that in a second. So I just want to kind of show you this bit here. So you'll have to label this kind of diagram, so this is an electron micrograph, so you'll need to say what band is what. So, I is actin, that thin filament, so it appears lighter, so kind of white colour. H is just myosin, just the thick, so that'll appear like a pale grey colour. And the A band is both of them where they overlap, so that'll appear much blacker in colour because it's got two different ones in there. And then you've got the Z line, which is the two ends, and that's called the sarcomere between the two Z lines. And in the centre, you've got the M line. So when it asks you to describe the electron micrographic structure of striated muscle, you'll have to label a diagram such as this and say what different fibres make it up.
Just labeling.
You'll have to. It's probably just going to be kind of, if you describe it, you'll say that bit there is the I band that contains only actin.
Right.
That bit there is the H zone that contains only myosin. And it'll be an image, either like a diagram like that, or you'll probably get an image like that from an electron microscope. So that would be the I band, that would be the H zone, that would be the A band, Z line, M line. So I have labelled it all on the A3 sheet for you, so you have got it there. How are we feeling about that? It's going to keep building up, so please shout at me at any point. It should all make sense at the end. Fingers crossed. It's just random letters. I can't remember what language they were. German's coming to my head, but I don't think it's German. But the A, M, I, H come from another language. I think the I literally means brighter, stains brighter. So that's kind of describing the electron micrographic structure of striated muscle. And then you've obviously got mitochondria as well. Yeah. You'll miss the hard bit. We will go over it in revision next week. That's alright.
Laura, just to double check, is this H zone and
H zone and A band.
okay?